Thank you, Bob. Welcome, everyone. I'm Guilin. I'm the Associate Director of the Yale Center for Genome Analysis. And as many of you might know that we're at the West Campus, other than the medical school area. But it's pretty convenient, actually, if you want to go to our facility. And you can take the shuttle or you can drive. We offer, there's a free parking over there and plenty of space if you like to drive. And we don't have a pointer here, but this is our building picture outside the building and inside. And we have a whole live picture here. And okay, and this is our center director, Shunkang Mani, and this is me. And we also have Dr. Kaya. He's the associate director for the sequencing section. And today we also have Chris here. And if you have any question you want to ask, we all very happy to help. And if you want to go to our facility and have a look, thank you so much. And we're happy to give you a tour inside our lab. We have a pretty amazing instrument inside of this building. And we are very proud of having those instruments to be able to help the research here at Yale Cancer Center. So what we do, we offer two major genomic analyses actually from our center. One is the microarray, and the other one is the next-gen sequencing. And pretty much we offer the services from either DNA or RNA. We even offer the isolation of DNA from or RNA from the tissue or the cancer tissue you might have, and also for the blood. After we getting the samples from you, we would prepare the samples until they're ready for the either microarray or the next-gen sequencing to be able to put on those instruments to get the data. Most of the time would be the primary data out of the instrument and send the data to you. And we kind of cover a lot of area using those two technologies from the gene expression detection, between cancer and control case, and also the full transcriptome analysis using both microarray and next-gen sequencing. And one of the major, I think, application using our center is the whole genome, whole exome sequencing, actually. Basically, you targeted the exome region of the whole, the human genome and put it on and prepare the sample and put on the sequencer, and would give you the insertion, deletion, SNP informations, out of the sample you might have. And now we start to offer the whole genome sequencing. By that, you not only be able to look at the exome region of the genome, which only 2% of the whole genome. You can also look all the other non-coding regions, like the long non-coding RNA region and all the other regions not covered by the exons. And we also offer microarray analysis. This is the, we have the Illumina and the Affymetrix array. They're covered the whole genome, all the known SNPs you can detect in a single one array. And one unique thing about the microarray is you can do really good methylation detection analysis. That's so far very good at the investigating of the epigenetic change might be involved any of your research. So for Cancer Center, actually, we have one fully supported bioinformatics staff, Dr. Francesco Lopez. He's the bioinformatician. Since any of those genome data, you really need someone to be able to help you analyze the data. And he's the 100% supported by YCC, so it kind of can give you from the designing of your experiments to the data analysis all the way to get the data you're looking for. Like I said, we work closely with the YCC members, And if you need us to help you write a grant, like David mentioned, we would be happy to give you those letter of support and give you suggestions and on working on those grants. So, like I said, this is basically the powerhouse, all the instruments, major instruments we have to be able to perform all the analysis, process all the samples for you. And those are the microarray equipment, and those are for the Illumina array, and this is for the FA matrix array. And as you may know, that the microarray is kind of older technology, but it's still widely used so far. There are still very unique applications you can take advantage of. And the major advance for the microarray technology compared to the sequencing, I'm going to talk more later, is that the faster turnaround time. We normally would give you the data within 2 weeks once we have your sample, and cheaper price compared to the sequencing. And also the easy data analysis, actually, since there are a lot of available software tools you can use to analyze the microarray data. But the real powerhouse, actually, are those guys. Those are the next-gen sequencing instrument, and those 3 are from the Illumina, and the other one is from the Pacific Biosciences, and also one from the Iron Torrent, the PGM. All the sequencers, they have different unique applications you can use for. For the majority of our sequencing work in the center, the number 1 are those Illumina sequencers, basically. And there are some older versions, the most recent version, and a smaller benchtop version. All those can give you a huge amount of data with a very cheap price in a very short period of time. It's very good for the whole exome sequencing and also any of those targeted resequencing approach, RNA sequencing. And the other sequencer we have is kind of unique. Is the Pacbell RS2 sequencer. The sequencer is different as the Illumina sequencer to give you really short, like 200 base pair reads. But for the Pacbell sequencer, it actually can give you average 10 kb long reads, can go as long as 60 kb long reads. So the major advantage for this sequencer is to sequence the full length of the cDNA transcripts to be able to tell you the isoforms in your cancer samples, and also be able to solve some of the facing structures, those long-range structural changes by using this technology. And since they all generate a lot of data, so we do need a dedicated cluster. That's the high-performance computer infrastructure we have. This is the we call Bulldog N cluster. Any member can get an account with the Bulldog N and do the data analysis. There will be limited space and computing powers. But for majority of the work, if you want to run yourself, you could use this for the data analysis. So those are the areas, for the researchers at Yale as well as the YCC been doing. Those are the some of those research areas that use our services. We do offer the CLIA certified sample processing. Most of those are the whole exome sequencing samples, and also as well as the microRNA, DNA methylation detection, and like the most popular one, the genetic variation detection between groups and among populations. And we do do the de novo sequencing even for the human samples. And since the price is getting down dramatically for the past years, and also some other areas. But using our services, there are a lot of publications out of the using our center's next-gen sequencing services and the microarray services. And here definitely is a partial list. And as you see, there are a lot of high top-tier journals in the Science, Nature, and Science. And some of those publications are from all the cancer center members working on all different kind of cancer types. And with that, I would like to answer questions if you have any. All right, good luck.